Hey there, and welcome to minutebiology, and today we will be talking about why what you eat is what you reap. If you zoom in really close, past the tissues until you're able to see the cells of your body, inside the nucleus, there's a small set of instructions that identify you as a human, and most importantly, you as you. This is your DNA. Naturally, the DNA will have to be split at some points of a cell's life, not only for mitosis, but also for a process called protein synthesis. In this process, the DNA gets unwound by an enzyme called helicase, leaving two half strands of DNA. Another enzyme called RNA polymerase comes along, reads one of these strands and constructs a complementary strand called messenger RNA, or mRNA for short. And in this mRNA, thymine bases, which are the ones labeled as T, are exchanged for uracil bases in their sequence. These mRNA strands are then transported out of the nucleus to the ribosomes, which combine information from mRNA and small floating pieces around the cell in the cytoplasm called tRNA. These tRNA pieces house amino acids. The ribosomes then combine all of the amino acids to create polypeptide chains, to which these can fold to secondary structure and tertiary structure proteins. The results of these proteins can encapsulate a lot of who you are, such as your hair color, skin color, and eye color. If we go back to your DNA and zoom out a little bit, you'll be able to see these structures. These are called epigenomes. These control what proteins get synthesized, or to put in another term, these control what genes produce their own protein from your DNA by turning on or off the genes. There are two main types of epigenomes, histones and DNA methylation. Histones are solid protein structures that the DNA wraps around. These structures are able to turn on and off genes by preventing the helicase from unwinding that area. For DNA methylation, these molecular structures called methyl groups protrude from the backbone of the DNA strand, and these are also able to turn on and off genes by preventing the RNA polymerase from reading the target area. Now let's take a step back and peer at the bigger picture here. As living human beings, we need energy, and we obtain energy by consuming luscious, delicious food. However, there's more to food than there is to that nice taste.
In 2018, a study was carried out on mice to determine whether the mother's diet can alter the epigenetics of the offspring. But why mice? Mice were used as they have such an anatomical, physiological and genetic similarity to humans. They fed the mother mice high-fat and low-fat diets and closely analysed their offspring. The mothers fed a high-fat diet produced offspring that had a heavier body weight than that of the mothers fed a low-fat diet. This means that the offspring were more likely to be fat. Not only did they give birth to obese offspring, but also those with denser bones. Differentially methylated regions, or DMRs, are also affected by maternal diet, as well as gene expression. DMRs are regions in the genome with different DNA methylation status amongst multiple samples. They are seen as functional regions that are involved in the regulation of transcription. Now, let's take a look on how micro and macronutrients can affect a child during gestation. Micronutrients are things such as vitamins and minerals. Macronutrients are the foods we know we need in our diet, like fats, carbohydrates, and protein. Too little protein, which is common in vegetarian and vegan diets, can lead to a certain mineral deficiencies, like iron or vitamin B12, causing a low birth weight or inadequate brain development. Fibre is another thing that affects a fetus. Fibre comes from plants and is very important for a healthy diet as it regulates gut movement. A high fibre diet is shown to prevent asthma in offspring, deeming it an important part in a mother's diet. Overall, there is strong evidence to suggest that a mother's diet has an effect on the offspring and their epigenetics, so remember to be mindful about what you eat because it matters more than you think.
I decided to look at how epigenetics and forensics work together. Within my research, I found that epigenetics is a relatively new branch of forensics and is not quite refined yet. Epigenetics tells the researcher or us what some aspects of our daily lifestyle is like, including how old we are, what kind of diet you have, how much physical activity you do, how much alcohol you drink, if you use any drugs and how many, if you smoke, and what facial features you may have. This is known as the epigenetic fingerprint. To gain the results, you would need to do tests in DNA methylation, but these tests vary and some can take up to a lot of time and may be very costly. The technology available is a limiting factor as this isn't accurate in some experiments. So as technology advances, we will be able to solve more crimes quicker and at a less of a cost. This is also why right now, the results from the methylation tests are more left up to debate rather than a scientific answer. There are also legal, ethical and social problems within forensic epigenetics. Since epigenetics can't tell anyone about a person's lifestyle, this could be a very personal to individuals or their families and exploit them to the public eye if it was to be published. This is seen as non-sensitive behavioural traits. The epigenetic information is also not stored within the police, as to not cause any bias within the cases. Epigenetics and forensics is a new strain of forensics, and it isn't as scientific as matching a fingerprint off a vase to the suspect's fingerprint. It is more down to your interpretation and to what scientists think the results would say about the suspect.